This evening's talk, we're going to find out about plant-eating beetles. They number over 120,000 species and form one of the largest radiations on Earth. Their ancient relationship with plants was crucial to their evolutionary success. Among them, the leaf beetle subfamily, the Cassidinae, known as the tortoise beetles and leaf mining beetles, offer a fascinating window into understanding this evolutionary history. The varied way of consuming plants has led to a wide range of morphologies, especially in their larvae. Different plant microhabitats have imposed unique challenges, and the beetles have responded with diverse defense mechanisms, including peculiar fecal constructions and even maternal care. And we're going to hear about this this evening from Caroline Chaboo, who grew up in Trinidad and Tobago, where her love for nature developed near rainforests and the sea. Her research into the evolution of the Cassidine beetles involves fieldwork all over the world to study species life cycles and to build up museum collections. She is currently a research associate at the University of Nebraska State Museum in the US, and we're delighted that she can join us here this evening. So thank you very much. Over to you, Caroline.
Thank you very much for inviting me, and for all of you out there who are viewing this talk. I will try to share my screen now. So this is a tortoise beetle. These are spectacular, beautiful beetles. Some of you may know them as, tortoise beetles, that's the cassidines, but not all cassidines are tortoise beetles. So we will get to the answer to that conundrum by the end of this 50 minutes or so. If you have questions, please contact me at my website. Okay. So they are beautiful, multicolored, any kind of colorful pattern you can imagine, polka dots, stripes, impressed elytra, lots of sculpturing. Unfortunately, some of these colors get lost in the dead specimen. So they lose some of these metallic colors, especially, which are due to water in the elytra. The cassidines share one common feature. And here you can see the underside of one where the mouth parts are sort of hidden. And that's they're referred to commonly within Chrysomelids, the family Chrysomelidae, as cryptostomata because of this hidden mouth. So let me just backtrack a little to something most of us know about or we appreciate. We're all steeped in a certain amount of knowledge about biodiversity on Earth. 20 years ago, when this figure was made, the number of described species was just over 900,000. But 20 years later, you know, we've gone up about 60,000 species, of which still we know that 1/3 of these are beetles. And we're looking at a long period of history, going back to about 400 million years, the Silurian, looking at fossils and all the existing species today to try to explain this incredible diversity in insects and in beetles. Why are there so many insects? When we look back, we sort of guess. We're kind of in a guessing game, trying to come up with ideas. We think, you know, it's because of their bodies covered by this exoskeleton. It's because of the wings that allow them to fly and pursue food and to escape predators. We think it's because some beetles can fold their wings backwards, you know, 99 and a huge portion of insects can do that. But it's remarkable that even within insects, this huge radiation of almost 1 million species, that are still a few groups dominate. And the most dominant are beetles. Again, looking retrospectively, why are beetles so diverse? And this has been really a wonderful question. It's challenged a few major labs around the world to look at this problem, sampling maybe 1% of the species to come up with these phylogenies, these evolutionary histories, to try and zoom in on an answer to explain why beetles are so diverse. So today we think that beetles are diverse because of their heavy body, their lifestyle with a pupae like some other insects. They have an unusual water controlling mechanism, cryptonephidia, but that's not so sexy. One of the sexiest explanations for why beetles are so diverse is their relationship with plants, because beetle diversity also happens to be dominated by these phytophaga, these few families that specialize on plants. So it includes the chrysomelidoids, the weevils, and the cerambycids. Another top-ranking major radiation is in the rove beetles, the staphylinids. And there, when we look retrospectively, we think that it's because they have lost some of that heavy body armor and they can move around and they're much more flexible. They can get into tight spaces and they're predators. They're active hunters. So we're kind of still guessing because we can't go back in time and see what happened. What was the tempo? What was the mode? How did this radiation happen? We can only look at what we have in museum collections today, counting species to know that, yes, these are facts. The life on Earth is dominated by insects, and insects are dominated by beetles, and beetles are dominated by these few families. One of these families are the leaf beetles, the Chrysomelids. This is a group I specialize on. It's too huge a group for one person to specialize on. So really, anyone who's working on this group, we all have to kind of choose a subfamily. But in general, it's a fantastic group, super diverse. As adults, with their colors, to find them, you really have to get to know some of the host plants, because you have to hunt, in the habitats for particular plants to find them. You use a beating sheet to collect them. We have a few phylogenies of chrysomelids, again, trying to explain this huge radiation. Today, we recognize about 12 to 13 subfamilies within chrysomelids, and we have a few proposals on the table about what mediated this massive radiation. It's plants, plant chemicals, or recently microbes. We will talk about cassidines mainly today. That's what I'm a specialist of. But you can see going around from tree to tree here, circled in red, it's really not fixed where they are. It's not really certain what is their sister group. It's pretty important to know your sister, who you're most related to start understanding the internal patterns. So we still have a lot of work to do at this level. It's a bit embarrassing. It's a challenge. It's a hopeful challenge. I hope more labs will get involved. Within chrysomelids, the diversity is very uneven, and cassidines ranks currently as the third largest subfamily with about 6,000 described species. So since this is a Linnean society, let's start with what Linnaeus did. With Linnean concepts, he described Cassida, and the name implying this metal helmet. And there's even today a brand, a patented motorcycle helmet called Cassida, which I love. Well, these beetles, as you can see there in the adults, the elytral margins are expanded. And so that forms this kind of tortoise-like morphology. The adults are rounded, the margins are smooth, the head is hidden in dorsal view. The larvae are exophagous. That means they eat outside the leaves. The leaves, they scrape or bite from the outside of the leaves. And they have these lateral projections as well as a paired hind pair caudal processes, which entomologists would normally call urogomphy, but in cassidines we call them caudal process, because we want to make sure that they're homologous, that we are referring to the same thing as coming from a shared common ancestor. The Hispa is the other group that you would that are included in cassidines, but they were not in the past. So 25 years ago, they were regarded as a completely separate subfamily, the Hispinae. And Hispa comes from the name Hispidus, from the word which means rough, hairy, or bristly. And as you can see in the European species that Linnaeus was looking at, they do have a lot of spines. But when you open your lens a bit wider, worldwide, not all of them are spiny, but they do tend to be this narrow, elongate form with the head partly hidden in that prothorax. And the larvae, what we thought 25 years ago, were these elongate, simple forms. And I put simple in quotation marks here because this is a microhabitat to live inside a leaf that heavily, severely constrains morphology. So in fact, these simplified forms are convergent across insects. A lot of mining insects, mining larvae tend to look alike. So it's a huge problem when you are looking at a leaf, trying to understand who is living inside and what even the order is. It's a big problem. So the fundamental problems about 20 plus years ago when I was starting out my PhD was that there were these two separate subfamilies, and then there was this group that was kind of in between. These are cassidines that are exophagous, but they live in these cryptic habitats within the bracts or the leaf rolls of zingiberelles, of heliconia type plants. I won't go down that route today with these Phytophagan cassis, but I recommend looking at the works of these two other researchers their labs have specialized and they've done really wonderful work on this group of cassidines. So the cryptostomes, to just recap, the big problems have been how are these two groups related? We're looking at more or less similar diversities. And then the higher level problem is how do they connect to the rest of Chrysomelids, the higher tree of life of Chrysomelids? What is the sister group? So this is where I want to start. When I graduated with my PhD, this was a monograph that I published on this group that made some progress towards resolving those initial issues of what is a cassidine, what a cassidine. So today we generally recognize Cassidinae as including these two groups that were previously recognized as separate. And they are connected through a whole suite of morphological features in the adults and some in the larvae and pupae, and because we don't know as much about the immature stages, a lot of characters on the tarsomeres, on the feet. So I'm confident today we're looking at a monophyletic group with a single ancestor way in the past. Now, I don't do catalog work, but this is a very important part of systematic research, which is the approach I take to looking at biodiversity. So I have to give a shout out to these two separated catalogs that I rely on and use pretty much daily. Not every group is fortunate to have catalogs, updated catalogs. And this is a key to learning the names of species. Sometimes you can use them to find the photographs of the species. It is a key to the literature. And we're looking at 400 years of literature, going back to Linnaeus that I have to trace for my group. So I wouldn't be able to do much of the work, even at the species level, if I didn't have these catalogs to rely on. What the phylogeny I built in my thesis, my PhD thesis, what it's now allowed me to do is identify new kinds of questions and develop new kinds of research. So now I can go a bit further in researching these beetles. I can go more deeply into trying to understand their biology. With the systematic part, I want to keep challenging all concepts of groups, of tribes, genera, of the subfamily, but we also need to look at the monophyly of those tribes to make sure they're natural groups so we can actually kind of understand better the biology we are observing. the evolutionary patterns now become more in my focus because I now have a lot more experience with the group, living and dead, and more familiarity with the species, and I understand a bit more of what's going on morphologically and evolutionarily. So this allows me to discern patterns that were not so obvious before. So phylogeny is a kind of a treasure map. It sums up everything we know, and then it allows us to strategize and go forward with new research. When I came off the PhD, I got a job as an assistant professor, and I also had to integrate teaching, which I love, with my students, especially in fieldwork. I also found that I was a little bit tired of traveling all over the world. And when you're traveling with groups and with students, you need to ensure you have local relationships to rely on if something goes on, to help set up the fieldwork, for permits. And I also wanted to learn a lot more about the biodiversity of one's place. I picked Peru, one of the most hyper-diverse countries in the world. So I probably need a few lifetimes to do research there, but I go there annually with students, and we do intense inventories of beetles. And then the period that we're staying there allows me to do some of the biological observations of live animals that I can't do here in the United States. So I work in the south part of Peru on an Andean Amazon transect from Cusco down to the Amazon. This is a view from one of my field stations. And when you drive down, it's just a spectacular view. From the grasslands all the way down to the Amazon, you can find cassidines. So they're very diverse in their habitat range. We use all kinds of traps. These are general traps that we install in the canopy, on the floor, mid-level, and they capture many things. So we end up with a lot of material, not just beetles, not just cassidines. But it's a good way to inventory a place objectively. And every year I go, I randomize the placement of these traps, and we place them in different areas along this transect. So we're using all kinds of traps, and then when we come back with this massive amount of material, again, I'm relying on a lot of students to help process these specimens. We can train them to initially sort by orders, to pin, to prepare them for the collection, to digitize. We cannot underestimate the amount of work it takes. It's great to go off on fieldwork, and it's really fun and exciting to go travel. But when you come back, the real hard work comes on. So kudos to museum people for the effort and care that they put into collections. So in the Peru beetle inventory, I've now documented over 101 families of beetles, and those specimens have been sent off to many experts all over the world for further study. The top 5 beetle families in Peru mirror the global diversity. And I didn't start out trying to prove that Chrysomelidae are number 1. It's not my interest to be the biggest group of all, but at present, that is the top most diverse beetle family in Peru. Within Chrysomelidae, again, I was shocked. It's not something I was hoping for. I didn't anticipate. Cassidines are the most diverse group, and this is completely bizarre when you look at the diversities of other groups. So we need more people on the ground. We need more inventories in different parts of Peru, and we need more experts, because I can take in other families things down to genera in some cases, or to tribes, but I need more people to take things down to species. What this fieldwork allowed me to do in Peru really is spend time identifying my host plants and getting specimens and rearing life cycles. So this really is the main purpose of my fieldwork, but it's what I hide inside all the other things I'm doing, because I'm doing this very basic thing, very old-fashioned in a way, of collecting larvae of their host plants, keeping them in containers. It's not an expensive, big science project, but every life cycle I collect is enriching specimen collections and giving me a lot of data, more data than I can handle, process, or even understand or write up. So I'll introduce you to some of the different Cassidine life cycles that I've looked at that can tell you something about the diversity within even this small group of 6,000 species. There are many ways to eat leaves. There are many ways to eat plants. And these beetles specialize on all parts of plants. Even in the rainforest canopy, we can find beetles that are drinking from extrafloral nectaries. When you look at mining insects, as I said before, it's really hard to know who's there. I have more experience now. When I know the host plant, I can suspect who's there. I can go look. I can keep revisiting that plant on my trail walks in a habitat. And then ultimately I can come back, return home with the life cycle, with stages of the eggs, the different larvae and pupae, and the adults. I absolutely need the adults to be able to identify the juvenile stages. Some of these are quite large, and some of them specialize on palms and grasses, the exophagus. This is where you can see really the pest aspect of chrysomelids. They really can severely damage plants and fruits and flowers. Some of the biggest pests, agricultural pests in the world, are chrysomelids, like your potato beetles. This one is a pest of palms, oil palms. This group in Australia, we are waiting to be accepted for publication. It's been a really fascinating tribe and genus to look at because they're exophagous, but they look so much like those basal hispine forms. They narrow, elongate body, but in this case, they also have this caudal process, this urogomphus. It's a really amazing group. And then there's the Phytophagan species. These live in bracts. I'm sure you know these plants as horticultural specimens, and you buy them at flower shops. They live both in the bracts and leaf rolls. So I can cut the bract and dissect it and collect the specimens inside those little pools. Illustrated here, you can see it's a whole mini ecosystem that's in there. And then there are the leaf rolls, and you unfold it, you open it up like a wrapped present, and there are all these cassidines in there, along with their larvae. It's really hard to identify these species, to associate species of larvae with the adults, because some of these life cycles are like 2 years, and I can't get the grant money or convince anybody to send me someplace for 2 years just to do that. But they're extremely weird. This research angle opened up a whole new world for me to be able to teach undergrads, introducing them to ecology and ecosystems and invasion biology. It's been fascinating, and we've published quite several papers at this point. The typical tortoise beetle is like this one from South Africa, where you would, if you look especially on the underside of the leaves, you will find the larvae, and if you are vigilant, you might find eggs, and then you can bring them indoors into containers and give them fresh leaves daily, and you will get the life cycle. In a tropical habitat, if you're in the right time, right when the rainfalls start and leaves are flush, you might get all the life stages within a 3 to 4 week cycle. In other places, it might take longer. I can't be every place all at once, and I am been blessed to have fabulous collaborators all over the world. It's a bit of a, they don't realize when they contact me with a picture, hey, can you identify this? That I'm just reeling them in little by little, because sometimes these non-Cassidine people, these naturalists, have such a great eye for the animals that it's easy to give them a list of questions and a how-to, and next thing you know, we are working on a paper. And this is the case with several of my collaborators who are non-formally trained scientists, but they have a great naturalist eye. Sally Adam in Cape Town is a sheep farmer. She makes cheese for a living, and here she had these beetles on her farm, and we were able to do this great project. This species has been introduced in other countries, particularly Australia, to control the weed, the host plant that came in as a garden plant, and now it's become invasive. So cassidines can be good guys, to be pests, economically damaging our food crops and our plants, but also they can be, biocontrols because they can be so specific. If you know a few of the most common families like Ipomea, morning glory, the convolvulaceae, you can find them easily, in the right habitat. So at this point, I have a large collection of baby cassidines. I feel very proud of that because the majority or nearly all museums that have insect collections have pinned adults. Very rarely do they have larvae. It might be even less than 1% of the collection. And it's kind of, it's really embarrassing and shocking to me when you think, how much of human biology do we know based on the immature stages? We really need to know a lot more about juveniles to understand the whole life cycle and to understand the evolution of these groups. So going back to this old phylogeny, and I just want to warn you, the only reason I'm using the old phylogeny, although we have some new phylogenies in the work, is that the basic topology doesn't change. The layout is about the same. We are tweaking a node here, a branch here, the definition of something here and there. But the basic pattern and the sequence that I'm using as the guidepost for this talk really hasn't changed. So we know now that the basal cassidines, the formerly hispines, are much more interesting than just being mining insects. They show a lot more variability and how many times they have emerged to feed on the surface. With the exophagous ones, the ones that became permanently exophagous, the tortoise beetles, I've been lucky in this transition node to have two fossils, to describe two fossils. And here, it takes a little while to get your eyes to look at fossils, to see what you're seeing, to understand what you're seeing. But here you can still, after a while, you can see the pattern emerge from the rocks, and you can see that hidden head. You can see the antennae that are close together, just like a cryptostome. You can see the explanate margin. And we were able to place them in this node, which is extremely important in this hinge point between basal cassidines and derived cassidines. So at this point, I will change gears a bit to focus now on two patterns of exophagy in general. It's a very dangerous environment to live on the surface of a leaf. So within all of Chrysomelidae, it's really peculiar that cassidines have gone inside a leaf. In brucinidae seed weevils, those chrysomelids have gone inside seeds. So it's escape and radiate. They've escaped from the exophagous enemies and gone inside, but then within cassidines, they re-emerge again multiple times. And so what's going on here? It's fascinating. So living on a leaf, exposed, and if you're an adult, you're very colorful, you're exposed, has a lot of enemies, a lot of visual hunting predators. And the first thing in the past we would think about would be birds and lizards, but actually other insects are like the biggest predators of cassidines, especially ants. I really hate ants. They do such a vicious things to my beetles. The ants will really cut them off, but bugs, hemipterans, pentatomids, scutellarids, they will stab them and walk off. So within cassidines, we start seeing in the more recent part of the tree that some of the larvae started living together as a group, like a little herd. And there is protection in herds. They reduce their chances of being eaten. But the most derived cassidines also show this marvelous wonderful behavior. It's so easy to sell beetles when you can show them something like this, where the mother stays and guards the group. So within the phylogeny, I can see that grouping behavior of larvae originated first, and then maternal guarding was added on. And these larvae, they might be feeding scattered on a leaf, but if they sense an enemy, a predator, or a danger, they cluster together, and she comes and walks all over them. She will fend off her brood from attack. Some of these cassidines are sexually dimorphic, with the males that fight. And some, you see this on the right side are the males with these extended elytra, and they fight and they stab each other with those corners, those extended stabbing humeri of the elytra. We filmed this behavior in Panama, but at this point, the digital technology has changed so much that it looks quite grainy and unappealing when you look at old footage. So I'm hoping that this piece of film that we shot in Panama will be digitized. Here's another subsocial chrysomelid where the mom guards the baby for about 10 days, and then she nurtures all the instars until they pupate, and she stays with the pupae as well. So she guards her pupae, which are stuck there, and then she only leaves and feeds when her young adults disperse. So it's like, I'm a mom, when my kid goes to college, then I can leave, then I'll be a little bit freer. So within this phylogeny that I have, that is allowing me now to summarize and understand some of the patterns within this group, I can see that maternal guarding came very late. It's a very recent innovation in cassidines. It's very attractive, and it's the first thing I can tell people about cassidines, but it's a minor, it played a minor role in diversification. However, we don't know enough about the species biologies of these small genera, so it'd be really fabulous for a small army of people to focus their talents on this part of the clade, because there's so much to be done with the behavior here in cassidines. What this is allowing me now, when I look at something in my group in cassidines, it allows me to step back and take a broader picture of chrysomelids, or even of beetles or of insects. So I'm changing my lens back and forth between this focal group and trying to understand also patterns outside. Sometimes I need those outside patterns to help me develop and structure the research to look at my patterns. So one of these is that we could now really understand that subsociality also occurs in another subfamily of chrysomelids, in the Chrysomelidae. And again, you see the spectacular thing with these colorful larvae and adults. They live in groups. We don't have a big, deep phylogeny of Chrysomelidae, and hopefully this will grow. But we developed a small template to understand, again, the pattern. So again, it's like developing the baseline phylogenies that will help us structure future research. We summarize the patterns we know now, and we discover more, and we kind of develop the story from there. The most dominant macroevolutionary event in Cassidinae is related to this really bizarre defense, where the larvae carry around these structures on their hind end. They wave it around. And if you look at it from different angles, it has an extremely ornate structure. And if you use a pencil or in lieu of a predator, they will pull it down on their backs. So they use this shield, as we call it, as a protective mechanism. To study that, I really had to dive deep, spend a few years really learning about insect animal constructions. Right away, you can think about birds and spider webs, termite mounds. In North America, we have beaver dams, wasp nests. The research on those in those taxa are much more advanced, but they give me clues about how to approach and develop my mode of researching this bizarre behavior in cassidines. I also focused on insects themselves, just insects, solitary insects that build structures. This is a assassin bug with it's collecting all the corpses of its victims, the ants. This is a lepidoptera that is cutting twigs and piling it on and building this amazing structure. And then we've co-opted some of these behaviors for art, for jewelry, these Trichoptera caddisflies. If you provide them with semi-precious stones, they will build you their cases when even in the aquaria that they live in, when you create an aquarium. It's really marvelous. I find this a complete black hole to go down, and I may never come back out, because all these little individual builders, they create such amazing structures for such diverse purposes. Some of these are houses, some are traps, some are nurseries, hiding places. Some of these animals live inside them and can sneak up on their prey. They can camouflage, disguise. In cassidines, they use a really fascinating material. They use their own poop. They're not like dung beetles that are rolling other animals' poop. They use their own. And if they're feeding well, they're producing it copiously. And the diversity is just astonishing. These larvae have this mobile structure on the hind end, and they're walking around with weight on their back. So evolution is economical, so it's not going to produce some fanciful structure that has no purpose. In the groups that live together, it can look quite bizarre. You don't really understand what you're looking at. And if you try to probe them with a pencil or a forceps, they will start flicking together. So they move it in motion. So they create a group behavior to make themselves larger than life. So the architecture of these structures, the different ways to shape poop, their feces, is really, fascinating. And if you visited me, I'll be happy to show you them under a microscope, what they look like. So to build a poop shield, you really need a unique kind of anus. And I don't think there's any other animal group that has such a unique anus as these guys. They have a flexible telescopic anus that moves around. And if this shield is broken or part falls off, they can repair it, and they can stretch that telescopic anus all the way up to the pronotum to repair that shield. So that's one key element of this construction, is the anus. My boss in New York, David Grimaldi, used to tease me that I had become a real systematist when I was counting the hairs on the anus of these beetles. I can laugh, but it's really something else to watch these animals. I want to show you a film now that it's 2 minutes long, and I'll try to narrate. It's part of a Japanese film series filmed with my collaborator in Costa Rica, Kenji Nishida. I've used some of his pictures in here. The second element is the materials. Now you think of the feces as the primary element, but there's a second element in these shields, and it's the exuviae. As you know, insects cast their skins for each time they want to grow to the next stage. And the skin can be just left behind, attached to the plant, or they fall off. But in this particular beetle, in these tortoise beetles, they retain that exuvial skin. This film is sped up. It takes about 20 minutes in real life, but it's sped up to 2 minutes. So just bear with me while be fascinated with me. It looks violent, but it's not, because it's sped up. So that happens at each molt into the next instar. Baby larvae have, in cassidines, the number of larvae can range from 3 to 9. But in this particular tribe, there are 6 stages, 6 larval stages. And each time they go through that process, they keep this exuviae and they walk around with this kind of shield. So this shield has no feces on it. And so there's diversity in many ways with these shields. All the shield building cassidines have the telescopic anus, but not all of them keep shields, and not all of them have feces. And when you dissect the structure, you can see how they're interlocked. They use the caudal processes, those urogompi. They become the central scaffold of these shields. The caudal processes themselves also have a lot of diversity. They can be usually commonly paired, but you can also have a single one. But this single one, I find it on a tree, on a phylogeny in the basal cassidines and also within the derived cassidines. So it can be gained, it can be lost, it can be formed. It can originate as a singleton, or it can be formed later on as paired caudal processes fuse. So it's complicated. So if you're just looking at a shield, you may not appreciate this elaborate morphology that's associated underneath. How does this whole thing work? Now, I can do my little experiments to figure out how it's working, but I'm relying really on many people who for the last 20 years have tackled this question of how these things work and why they have it, to bring together the data. Because when you're looking at it in an experimental way, you are usually studying only one species, whereas I can compare many species. So I have a kind of big umbrella view, but I am relying on all these other researchers who are looking at individual species. And there are a whole lot of people. It's a very interesting set of data that has come together at this point. So if this cartoon of a baby larva, they're keeping the exuvia, and the exuvia becomes the central stack, and then they're using the anus to build these fecal filaments or blobs or wet feces in different ways. Now, new data is emerging that suggests that this shield, besides being a physical barrier that protects the animal and that may be offensive just because of the material that it's composed of or being a camouflage, may actually have chemicals in it. And these could be from the gut, from the plant, based on what they've eaten, or also from microbes. We don't know. So, and when I look at shields, I've been able to find fungi growing in the shields. So this may be a whole garden that they've cultivated that they keep on their bodies. Certainly, we know that if you remove the shield, they will be picked off by predators and die. So we have enough data to show that this is an important evolutionary novelty. We have this huge radiation of cassidines, about 3,000 species that show this structure, and it's correlated with the sophisticated morphology of a telescopic anus and the caudal processes. Evolution is economical. It's not going to produce anything fanciful, excessive, that we think of as excessive, that doesn't have a function, that doesn't have a utility. And certainly there are costs and benefits to carrying around this weight, and to producing it and investing in the morphological equipment to build it. So we have enough data to show that it's serving a very important function in the life of the beetle, and so we can infer that it had a huge impact on cassidines because of this huge radiation after the origin of the structure. So the basal cassidines don't have it. It only comes at that hinge point and subsequent radiation. This now, again, I'm going to toggle back and forth. I was looking at insect constructions through all of Animalia. Then I narrowed my lens to single individuals that build, single insect individuals that build. Now with this story within Cassidines that I have some understanding of, I can widen my lens a little bit to say, well, what's going on in the rest of Chrysomelids? And it turns out that fecal recycling occurs in several other branches, in several other families of Chrysomelids. Within the Camptosomata, these are two subfamilies, it's even more complicated. Here, the mother initially builds a fecal case around the egg that the larvae inherit and expand and build and retain. And when they're ready to become a pupa, they seal it to the substrate and they pupate within this fecal construction. And they walk around with this very heavy structure. And they're quite hard, so you can keep them in an alcoholic collection. And it's a huge radiation of this group, 6,000 species, just like Cassidines. Another smaller lineage are the shining leaf beetles, the Chrysomelidae. And they don't have caudal processes. I can't find a morphology with it, but their anus is not telescopic, but it's a little pore right on the dorsum. It's not on the back end, it's not ventral, it's on the dorsum, and they poop on their backs, and somehow they build this armor right on their backs of wet feces. It can fall off, but they replace it all the time. A similar one is made in the Galluricines, but this is a minor lineage of about 200 species, the Blephariida group, and they keep a similar kind of coat on their backs. And this week, my collaborator in the Philippines is doing the experiments to clean the larvae so we can start seeing how they build. So the same kinds of parallel things I have done in Cassidine to understand how they build and what they build it with, I'm trying to now replicate in these other groups. This is an interesting group, and I'll just drop it in here because some of you may be familiar with my work. These are the group from which we get the arrow poison beetles. These are the ones in Southern Africa that the sand bushmen collect, and they use the pupae. They collect the cocoon underneath the soil in the Kalahari sands. They dig them up, and then they break that sand-based cocoon, and they take the larvae out, the last instar, not the pupae, the instar, the larvae, and they use that to tip their arrows. Now, I've done field work in South Africa, Namibia, Botswana, and all the different sand groups using different species in their different habitats. So what I've uncovered here with Cassidines is that there's this huge macroevolutionary event within Cassidines with this fecal construction and the associated morphology, and it's beneficial to the animal. Across Chrysomelidae, we now have uncovered 5 branches with fecal recycling, and they build different kinds of structures. They appear as independent events, but the idea that they are all within Chrysomelids suggests some kind of underlying genetic architecture that is governing this behavior. The 2 major radiations of fecal behavior, fecal recycling, are in the Cassidines and the Cryptostomata, 2 groups of about 6,000 species each. The other 2 lineages, the Chrysomelidae and the Blephariida group, are minor. So at this point, in a kind of summary, is I have this phylogeny that has been my map, guiding my research, summarizing what was known, and leading me to search for new things, to uncover the biology of individual species and try to pull all of that into this framework. Phylogenies are really marvelous tools for summarizing information, biological information, and allowing us to discern patterns that we didn't really recognize before. So my next step, probably for the next 10 years, is really to kind of wallow in this hinge point within the tree. I've laid down the first wave of publications with the basic natural history. I've identified field sites. In 2 weeks, I'll be in Trinidad visiting my parents, but really undercover to look at Delocraniini, where I found some palm trees with this species. You need a ladder to get to these cassidines. But I'm really excited about this hinge point. I can focus on that one and feel I can do something constructive. However, the field is wide open for other researchers to come in, and particularly we need people working at the node with cassidines. Sorry, I have to figure out how I can. So at this point, I will take questions. I thank you all very, very much for allowing me to speak at you about these fascinating beetles and their weird behaviors. I have just had a lot of fun with these beetles, and it's great to talk to people about them, because when you're dealing with each individual paper, you're focused on that one thing, and giving a broader perspective, a broader talk allows you to put it all together and to think again, wow, maybe I made a difference, maybe I didn't, but I hope you found some really interesting ideas in this talk. Thank you.
Thank you so much, Caroline. That was so fascinating. Such an amazing group of beetles, from those beautiful adults to those absolutely so fascinating larvae, and what they're getting up to with their fecal shields. Absolutely amazing. So we have some questions. So we have time to ask you a few questions now, if that's okay. And if you would like to ask a question for Caroline, can you put it into the Q&A now, please? So I'm going to start with a question from Ray Heaton, who says, extremely interesting, thank you very much, and asks if there is any evidence that the beetle poo shields contain toxins from the plants.
Yes, but the analyses have been done on a few species. I'm waiting for data from a chemist who is analyzing shields we have sent to him. So we had a grant from the US National Science Foundation specifically for that question. It allowed us to identify a few field sites and go pursue some species scattered across a tree, collect the shields, and we collect the shields into methanol. In parallel to that, Fred Wenzel, who is in Panama, has been analyzing a few species. So we have preliminary data to say, yes, there are plant chemicals in the shields, and there are also chemicals that maybe the beetle altered plant chemicals and made something new. So we have that evidence. We have not yet any evidence that microbes or gut biota are producing chemicals. But this will come as we fall through each trapdoor, analyzing subsets of questions.
Oh, thank you. And like staying with the idea of the shields, it's really interesting that some of the groups are using the fecal shields, but others are just using the exuviae. And some of those, like with the exuviae just sort of piled on top of each other, it looks like it would make them more conspicuous. So do you have any ideas or suggestions of how that is acting like a shield for them?
Well, it's mobile, so they can bring it down flat on their backs.
Oh, wow.
Yeah. So if you put a pencil, this is what I like to do when I show my students how it works. It will hit you with a shield. It will hit the pencil point or the forceps. So it's really working, and it has a 360-degree kind of movement, forward, front, on the back, to the side, and back. So it is conspicuous to our eye. Now, here's the thing with the chemosphere angle. We don't know if it's masking within the chemosphere of the plant. So it may be undetectable in a way to certain kinds of predators. With exuviae, we know they may have old glands with some residual chemicals. We don't know. But again, it's questions to start like listing and tackling.
Oh, interesting. So it's kind of like this mobile appendage that they can do things with. Interesting. And related sort of to that area as well, Francis Taylor says, could some of those shed skins also be acting kind of as a mimic of like mimicking a predator or an earwig in this case, or something like that?
We don't know. You don't really find too many things carrying around such big structures. There are other beetle larvae that have huge, relatively large caudal processes. But the caudal process is already something that's sticking out the back end. But now they've built this skyscraper on top of it. So it's hard to know. We're looking at these creatures and analyzing them with our human perspective, but we don't know how it may be perceived in the environment.
Cool. And another question from Francis. So do other insect families exhibit this fecal shield behavior?
I'm sorry, if other insects?
Other insect families, is it this subfamily specifically, or is there a more broad distribution?
So within Chrysomelids, there are 5 clades. They're the ones that build the case, and then there are the others that keep it right on the coat without any special structures. But that's within Chrysomelids. In the course of doing the research for this paper, I learned about one weevil from New Guinea that actually keeps feces. And there's another weevil in New York that builds something analogous. It makes a little lichen garden on its back. So it's growing things, So the overall effect is of a shield, but the materials are very different. There is one caterpillar in Australia where they also keep their cast skins, the exuvia. But it's very bizarre how they keep the head of the old skin stacked on their own head, on their living head. So they're wearing with like a top hat or many hats on their head. But these are, maybe it's because we haven't been looking, and when we find one species, we need to look at the whole genus. But there's just so much that's unknown about insect larvae that we can't tell how big is the pattern.
Thank you. And then a question about the maternal care from Lynette Strickland. So she says, wonderful talk. Do you notice any clustering in the geographic distributions of the species that display maternal care? For instance, do most of these species occur in particular countries, environments, or things like that?
So the behavior of clustering of the larvae is worldwide. That behavior, you can go to India, Australia, Africa, and find groups where the larvae are clustered with their shields. So Aspidomorpha may be the most renowned cassidine because it's such a big pest of sweet potato in Africa and Indonesia. And so there's a lot of work with that particular species. But in maternal care so far in cassidines, we only know it from the Americas, from Central and South America. So it's taxonomically constrained to 2 tribes, and it's geographically constrained to the neotropics. However, the Chrysomelidae, the other group that has maternal care, that one is showing it in Australia. So and neotropics. So the geographic constraint may simply reflect geological history, where this tribe emerged in the neotropics, and that's where the behavior originated. But for Chrysomelids, subsociality and maternal care is older and more widespread.
Cool. Well, thank you so much. I afraid that's all we have time for this evening for the questions. But I wanted to say a massive thank you for absolutely fascinating talk. Thank you so much.